So hello class, I will be discussing another topic which is related to bacteriology, so also related to the previous topic that we have discussed, okay, the isolation to pure culture. So this time around we will be having the colonial morphology. Okay, so that means we are to observe how the colonies would look like on an agar plate or even in a test tube agar, okay, after we have isolated. Okay, so in this case, I have here a sample problem. So an exudate is submitted in the lab and the medical technologist perform gram staining and culture and sensitivity and did the workup after 24 hours of incubation. So here's the result, okay, for gram stain. So there are many white blood cells and also moderate gram positive clusters, okay, and in chains, okay, and few gram negative bacilli. So this is moderate gram positive cocci in clusters and in chains, and few, okay, few gram negative bacilli. So sometimes class, the gram stain is requested together with culture and sensitivity because this is the initial test, okay, on the possible presence or the type of the microorganisms that can be found, okay, in the patient's specimen. Because remember, culture would usually take 18 to 24 hours. And of course, the doctors would want to initially determine what causes the infection of the patient. Is it a gram-positive microorganism? Is it a gram-negative microorganism? So they can already start the treatment as early as possible by just the result of the gram staining. Okay? And another thing in culture, okay, in BOP, okay, we can observe three types of colonies. Okay, so the first colonies, okay, they are beta hemolytic. Okay, so meaning to say there is clearing, okay, of the blood agar plate. So clear or complete hemolysis. So beta hemolytic, yellowish, white, and creamy and buttery looking. So if you are a medical technologist, okay, I know that you already have an idea on this morphologic characteristics being described, okay, for the first colony. So creamy and buttery looking. So that might, okay, that might speak of the presence of gram-positive cocci. And since you have in clusters, okay, or grape-like arrangement, probably, okay, that is staphylococci. But it's not yet conclusive because what you are given are just the gram stain result and the colonial morphology. So it's as if you are giving clue, okay, to the doctor of what possible microorganisms, okay, are being isolated. Okay, another thing for the second colonies, they are also beta hemolytic, but this time around, they are large, gray, and mucoid. In this case class, you have to remember that gram-negative microorganisms, but not all of them. Okay, not all of them. Gram-negative microorganisms, okay, are usually large, gray, and mucoid on blood agar plates. So that's another clue. Okay, large in size, mucoid in consistency, and gray in color. So that would speak of the gram-negative bacilli that the medtech detected in the gram staining result. Okay, another for the third colonies, it's the third colonies are non-hemolytic, but they are also large, gray, and mucoid. So probably that is again gram-negative microorganism. And aside from that, okay, colonies were also observed on McConkey agar. And in this case, you only have two colonies. Take note, in BAP, you have three colonies, but in MAC, you only have two colonies. Why is that so? Remember, McConkey agar is supportive for the growth of what? For the growth of gram-negative bacteria. Okay, so in this case, I have indicated earlier that when we see large gray and mucoid colonies, okay, that could speak, okay, of the presence of gram-negative microorganisms. So that's why in McConkey, you only have two colonies. One is a lactose fermenter and another one is a non-lactose fermenter. Okay, and another distinct description that was given here, it's the lactose fermenter is surrounded by pink precipitate, or there's a halo, okay, surrounding the lactose fermenter. Okay, so commonly, class, these are the things that you can encounter when you are doing the workup, okay, in bacteriology. Okay, so you submitted, or I mean the nurse or the patient submitted the specimen, and of course, again, the specimen might contain numerous microorganisms. Okay, that's why you need to perform isolation technique in order for you to really isolate a pure culture. And by obtaining a pure culture, you can correctly identify the microorganisms present in the specimen of the patient. Okay, so aside from the gram stain result, you can have initially a clue of what microorganisms you are possibly identifying. Okay, another thing, so what are the importance of colonial morphology, okay, as a diagnostic tool? So number one, it provides a presumptive. Again, colonial morphology is not conclusive, but it could tell you, it could give you an identification, I mean an idea, okay, of the possible microorganism that you are isolating, and that you are giving the physician or the doctor a clue, okay, of the possible microorganism that he or she is dealing with, okay, and of course the treatment that he or she should give to the patient. Okay, another thing, enhance the quality of patient care through rapid reporting of result, and of course increase the cost effectiveness of lab testing. Okay, take note, if you know correctly on how to properly identify the colonies, on how to work up them, you know the description, the usual, okay, of the colonial morphology, then you are helping the doctor to speed up, okay, the treatment of the patient by generating correct and reliable results. And of course, it increases the cost effectiveness of the laboratory because you are not wasting the materials, the agar, and all the culture media that you are using in the laboratory, because you are correlating what you are doing, okay, and you are identifying correctly the bacteria present in the patient sample. And another thing, it plays a significant role in QC or quality control, especially of automated procedures and commercial system. Take note, class, that although there are a lot of machines already in the laboratory, we are still using control microorganisms. I have mentioned in my previous discussion about stock culture. So we normally keep that stock culture either in a test tube or in a Petri dish to serve, okay, to serve as our reference if in case we are to compare the colonies on the agar, okay, after you isolated, okay, the bacteria from the patient sample, of course, to the control that you have kept in the laboratory, the stock culture that you have in the laboratory. Okay, so that's part of the quality control and quality assurance also in the laboratory. Okay, so what we term to the initial observation, okay, of the culture is plate reading. Okay, and also you have to consider the incubation time. And as what I have said, bacteria would tend to grow, okay, from 18 to 24 hours. So you can observe colonies already on a Petri dish, okay, containing that solidified agar. Okay, why is it that incubation time is important, particularly in our example? Okay, because some of the bacteria will change their morphology as you continue incubating the culture. So just like, for example, in the sample problem, we have a presumed, okay, or we have a result, okay, for gram stain, which is presumed to be Staphylococcus aureus, right? Because again, we have seen, okay, cocci in clusters, gram positive, okay? So you have to take note that incubation time is important for S. aureus because S. aureus, okay, the colonies of that appear smaller, okay, when the culture is still young, okay? And of course, okay, speaking of hemolysis of the S. aureus is beta hemolytic, right? So in younger colonies, the beta hemolysis of S. aureus is not that distinct as compared to the matured culture, which has reached the 24 hours. So that's why incubation time is important. Another thing, okay, the growth of organisms on the media should also be observed. Is it only growing in BAP, or is it growing both in BAP and in CUP, just like in our sample problem? So you have 3 colonies in blood agar plate, and you only have 2 colonies in Macon K. Okay? Because again, since you are using multiple or different agar, then that means you can, okay, initially identify the type of microorganisms that you are dealing with. Okay? So is it a selective or non-selective media? So you have to correctly, okay, determine, okay, the growth on these kinds of media. And example, I think I have mentioned this to you in the class. So like Haemophilus and Neisseria. Okay? Haemophilus and Neisseria, they grow only on chocolate agar, but not in BAP and not in MAC. Okay? Although they are bacterial microorganisms, they are considered as fastidious microorganisms. So they require special nutrients. So in the case of Haemophilus, for example, okay, it requires NAD. This one, the NAD, okay, the V factor, okay, for growth. And remember, the V factor can only be seen in the lysed RBCs. So take note, in the chocolate agar, the RBCs there, okay, are lysed because of the manner of the preparation of the culture medium. Okay, so that's why it is supportive for the growth of Haemophilus, but not, but the BAP is not supportive for the growth of Haemophilus because, again, the RBCs in BAP are not lysed. Okay, another thing, gram-negative rods are better described in Macon K, of course, okay, because remember, you can see lactose fermenters and non-lactose fermenters because in the first place, MAC is a selective medium for gram-negative, and also it's differential in the sense, again, that it can differentiate microorganisms based on their lactose fermenting capacity. Okay? Another thing, true hemolysis is not seen in chocolate agar. So that's why you have to compare and contrast the colonies, okay, of the bacteria, okay, in blood agar plate and of course in chocolate agar plate, because sometimes you might observe that the beta hemolysis in the blood agar plate can be seen as a greenish coloration on the chocolate agar plate, but you should take note that true hemolysis cannot be observed in the chocolate agar plate. And of course, again, lactose fermenters and non-lactose fermenters are easily detected on Macon K. Okay, so those are just few of the things that you have to consider while plate reading. Okay, another thing. So these are examples of the gross colony characteristics. So you have, again, hemolysis, the size, the edge or the margin, the elevation, the density, the color consistency, the pigment, and the odor. And of course, when you read the hemolysis, there should be, okay, sufficient, okay, light source. Okay, and of course, as what you all know, hemolysis can be observed in blood agar plate. Okay, blood agar plate, and this is caused by enzymatic, okay, or maybe by the toxins produced by the bacteria. Okay, like the Staph aureus can produce hemolysin that could lyse the RBCs on the blood agar plate. That's why S. aureus is beta hemolytic in the blood agar plate. And of course, as mentioned in the class, this is useful in the differentiation of streptococci and enterococci, especially when you do the workup for gram-positive microorganisms. Okay, another thing, okay, so again, speaking about streptococci, so you have here the differences between the growth of alpha hemolytic streptococci. Number one is Streptococcus pneumoniae, okay, and another one, the viridans streptococci. By merely observing the size of the Streptococcus pneumoniae and viridans streptococci, you can already have a clue on what streptococci, okay, you are isolating. So in this case, Streptococcus pneumoniae, okay, it's umbilicate, okay, and of course, larger colonies. But for viridans streptococci, it's umbonate. This is umbonate. This is umbilicate. Okay, it's depressed at the center. And another thing, the colonies of streptococcus or viridans streptococci are smaller as compared to the colonies of streptococcus pneumoniae. So also another thing, okay, you can observe greenish coloration on blood agar plates. So that is not a complete hemolysis. So that is just an alpha hemolysis. So a closer look for the colonies of streptococcus pneumoniae again. So it's umbilicate and again large colonies. So you have here the depressed center. Okay, another thing. Oops. Let me just have it again as a slideshow. Okay, so this one, it's a typical colonies of streptococcus pneumoniae, aside from the fact that you can see greenish coloration there because again of the hemolysis on blood agar plate. The colonies of streptococcus pneumoniae are usually mucoid. Okay, mucoid colonies, streptococcus pneumoniae. For viridans streptococci, okay, again, usually the colonies are pinpoint, smaller than streptococcus pneumoniae, and of course, okay, you have still the alpha hemolysis, greenish hemolysis. So that's viridans streptococci. Another one, so this is not a pure culture because as what you can see, you can observe multiple colonies. So some are large and some are smaller in size. So this one, it's larger and it's umbilicate. So that's atypical for streptococcus pneumoniae. This one smaller, okay, and umbonate, okay, that's again, okay, a possible, okay, characteristic of viridans streptococci. So those are again for alpha hemolytic streptococci. How about beta hemolytic streptococci? When you say beta hemolysis, it's a complete hemolysis. There is clearing, okay, around the colonies. So just like what you can observe here. Okay? So beta hemolytic streptococci, you have examples, Streptococcus pyogenes and Streptococcus agalactiae. And you also have this gram-positive Listeria monocytogenes. So that's a gram-positive bacillus. Okay? So differentiating your Streptococcus pyogenes and Streptococcus agalactiae, okay, is easier if you already know by heart, okay, the characteristics of the colonies of these microorganisms, aside from the fact that they are both beta hemolytic. So as what you can observe here, the colonies, okay, of Streptococcus pyogenes are pinpoint. Okay, so pinpoint and smaller as compared to the colonies of Streptococcus agalactiae. And because their colonies are smaller, you can, okay, observe large or wide zone of hemolysis, as opposed to the colonies of Streptococcus agalactiae, which are larger, of course. Actually, it's described as medium-sized colonies. I'm just saying larger because it's larger as compared to Streptococcus pyogenes. Okay, but the colonies of Streptococcus agalactiae are usually medium in size. And of course, because of the medium-sized colonies, okay, the hemolysis, okay, the beta hemolysis is smaller, okay, because the hemolysis is being covered by these medium-sized colonies. And of course, sometimes during initial culture, or maybe the colonies are still young, okay, the hemolysis are not yet that distinct. So you have to remove the colonies for you to see the hemolysis, the beta hemolysis. Okay? So those are again Streptococcus pyogenes and Streptococcus agalactiae. And I have mentioned earlier that the alpha and beta hemolysis on BOP, okay, should not be mistaken, okay, with the greenish coloration on chocolate agar, because the true hemolysis is not observed on chocolate agar. So that should not be mistaken as alpha hemolytic on chalk. Okay? Next one, this is also an example of the colonies of Streptococcus pyogenes. Again, they're pinpoint. Okay, pinpoint. They're very small. That's why you can see wide zone of clearing. Okay, wide zone of hemolysis. Okay, on the other hand, you have here, okay, the Streptococcus agalactiae. Okay, it's like a bullseye because you have the medium-sized colonies and then you have smaller zone of beta hemolysis also. Okay, this one, okay, sometimes you might be using a type of blood that is not really helping you observe the hemolysis. Like, for example, you just use human blood instead of using sheep's blood. So, of course, between the human blood and the sheep's blood, okay, the human blood cannot really give you, okay, the true hemolysis that you are looking for. Okay, but a sheep's blood, okay, could give you the type of hemolysis, and you can easily differentiate the type of hemolysis. Okay, because again, sheep's blood, okay, although it's more expensive, but it is a lot reliable. Okay, if ever you are comparing the hemolysis of the colonies of the microorganisms. Okay, aside from the hemolysis, okay, gross colony characteristic can also refer to size. So as what I have mentioned earlier, they can be large, they can be medium, small, or very, very small, known as pinpoint. Okay, and another thing that I have observed based on my experience in Bacta section is that the gram-negative bacilli have usually larger colonies as compared to gram-positive cocci. That's why in the initial sample problem that we have, okay, I have mentioned to you that when you see large gray and mucoid colonies, that is again an indication of the growth of gram-negative microorganisms. Okay, so this size varies between species and genus of the microorganism. Okay, another thing, okay, aside from size, you also have the margin or the edge. Okay, so the margin can be entire, so it's smooth. Okay, the edge is smooth, undulate or wavy. Okay, filamentous, just like a yeast cell. Yeast cells are usually filamentous, so they have that fit-like projections, or they have that spikes. Okay, or star-like colonies. Okay, those are an indication of the growth of yeast cells on the agar. Okay, so undulate, wavy. This one, do not mistake it with lobate. Okay, lobate, of course, you have a lot distinct lobes. Okay, but wavy can just be like this one. Okay, erose, it's serrated. Okay, curled. Okay, the entire colony, okay, are usually curled or wrinkled. Okay, so examples of that, Bacillus anthracis. So the Bacillus anthracis, when you pick up the colony and then you spread it on a glass slide, okay, and look at it under the microscope, you can see a medusa head colonial characteristics because of the interlacing. I don't know if that's the correct term, but the Bacillus anthracis are usually overlapping each other, okay, and you can observe medusa head colonies, okay, on the microscope. So it's again filamentous also, okay, when you check the colonies, the individual colonies. For Proteus, of course, what is distinct is a swarming characteristic. And sometimes if you want to isolate Neisseria gonorrhoeae, for example, and you use Thayer-Martin agar or, for example, New York agar, so you can make use of trimethoprim lactate, okay, to inhibit the swarming of Proteus. Okay, trimethoprim lactate. That's to inhibit, okay, the growth, the swarming of Proteus. Okay, this one, this is the colonies. These are the colonies of Bacillus anthracis. Okay, so as what you can see there in the agar plate, they are usually non-hemolytic. Okay, and aside from that, aside from a medusa head description under the microscope, you can see in the book sometimes it's referred to as dry ground glass surface. Okay, ground glass. Okay, the colonies are dry ground glass surface. Okay, and another thing, there's this comma projection. Okay, with the colonies because the edge, okay, is irregular. And again, when you pick up the colonies and then you check it under the microscope, you can see this one. Okay, so under the microscope, the edge of the colony, okay, is composed of, like, again, long and interlacing chains of bacilli. So resembling locks of matted hair. So that's why it's described as medusa head appearance of the colonies. Okay, also this one, so that's medusa head, so that is Bacillus anthracis. Okay, this one also, they are interlacing. Okay, the bacilli are interlacing. Another one, this is a type of colony growing in Mac concave. So the margin, okay, the edge is soft, smooth. I'm sorry for that. The edge is smooth, so that is what we call as entire. Okay, smooth or entire, or other term is even again, even. Okay, another one. How about this? How do you describe this one? Okay, this is a wavy type colonies. Okay, how about this one? Okay, I draw this or I illustrated this earlier as like this. Okay, just like this one. So this is, okay, lobate. Okay, how about this one? Okay, that is, what do you think? Okay, so they are serrated. So what's the other term for that one? Serrated or erose. Okay, so that's serrated or erose. How about this one? Okay, how can you describe that one? It's very obvious. Okay, of course, it's filamentous. Okay, how about this one? Okay, so when you zoom out the colonies, so what you can see, okay, it is curled. Okay, it is curled or it's wrinkled, so curled. Okay, how about this one? Okay, usually you can see this filamentous growth of colonies, and you have again that star-like projections. You have feet, okay, at the edge of the colony. So these are yeast cells growing on blood agar plate. Okay, so that's the typical, okay, colonial morphology of yeast cells. Okay, another one. What is this? Okay, so there it is. That's the swarming of Proteus. So that's why the colonies are often overlooked, because again the swarm covers the colonies. So that's why if you really want to isolate bacterial microorganism, to pick up the colonies and to prevent or to inhibit the swarming, again, the example that I have given you is the use of trimethoprim lactate. Okay. Also this one, so that is swarming. So you can see here swarms of Proteus. Okay, another thing you can commonly observe. So you have there the colonies, but again, there is swarming. Okay, so just like this one. Okay, so that's how you work up. So you're not just looking entirely, okay, at one colonial morphology, but you also have to take a look at the gram staining reaction. You can perform germ tube if ever you have isolated yeast cells. Okay, like for example, okay, if you can see yeast cells on blood agar plate, usually they are not pathogenic, except if your specimen is vaginal swab, or like vaginal culture, or of course blood specimen, because blood is sterile in the first place. So one of the clues that you can look for, okay, if you really want to identify candida or you really want to isolate candida, is again the feet projections, okay, at the edge of the colonies. Okay, so if it has feet, then it is suspected for Candida albicans. However, if it doesn't have feet, okay, it can be also Candida albicans, because other colonies would tend to, okay, have smooth margins, and they would look like the colonies of Staphylococcus aureus. Or maybe, okay, aside from Candida, it might be Torolopsis glabrata. Okay, it's another fungus. Okay, Torolopsis glabrata. Okay, but the difference is that the Torolopsis glabrata grows poorly on blood agar plate. And unlike your Candida albicans, okay, the colonies are very tiny on the first day. And take note of this one. Okay, gram-positive cocci or gram-positive bacteria grow, okay, on BAP, on chalk, but not on MAC, right? Gram-negative bacteria, on the other hand, okay, can grow on BAP, can grow on chalk or chocolate agar plate, and they can grow, okay, also in MAC concave. So if ever you have growth in all plates, like the BAP, the chalk, and the McConkey, that is already an indication that it is a gram-negative microorganism. But, okay, you also have to look exactly at the colonial morphology because that can also be a yeast cell or a fungus. Okay, because again, yeast can grow on McConkey. Okay, another thing, if you can see opaque white to yellow, so that can be suspected for staphylococci, especially if it is catalase positive. Okay, but you have to take note that it could also be a yeast if it is mucoid. It could be Cryptococcus neoformans if the colonies are white, or Rhodotorola if the colonies are pink. Okay, so they are often confused again with what? Staphylococci. Okay, and if you are really suspecting it as a fungus like Cryptococcus neoformans, okay, the fungus that infects, of course, our brain, you can perform India ink. So this is a very easy procedure. So you can just put a drop of CSF on a glass slide, and then you put a drop of India ink, okay, also on that CSF. And then you cover it with a cover slip, and then after a few minutes, you can examine it under the microscope and you can see halo surrounding the yeast. Here's the yeast. Okay, so that is, okay, again, conclusive for, okay, Cryptococcus neoformans. And of course, if you have not performed an India ink, but you have observed in the gram stain that there is a pink halo, okay, surrounding this yeast cell, pink halo, that's again in gram staining, okay, you suspect it as Cryptococcus. And another thing that could help you identify it as Cryptococcus neoformans is that Cryptococcus neoformans is urease positive. Okay, but again, conclusive, okay, we really want directly to identify the microorganism, you can directly perform, okay, India ink testing. And of course, the yeast cells look like this one, okay, in the gram staining. So you have here the hyphal elements and of course the budding yeast cells. And of course, if you suspect it to be Candida, okay, particularly Candida albicans, you can perform a germ tube test, okay, germ tube. So how do you do the germ tube test? Okay, you just mix the colony with a plasma, citrated plasma usually, and then you incubate, and after that you look under the microscope and you see, okay, the appearance or the development of germ tube. Okay, this is not a germ tube because in the middle there is a constriction here. You will know that it is a true germ tube if there is no constriction. But if it is like this one, okay, this is not a germ tube. Okay, that's just a pseudo hyphae formation. Okay, and of course, this is how, again, the yeast cells would look like under the microscope and when you perform gram staining. Okay, another thing, elevation. So you look at, okay, the colonies. Is it flat, raised, convex, pulvinate, or umbonate? Okay, so again, umbilicate, I have mentioned this one. Okay, umbilicate, do you remember, okay, the microorganism that is umbilicate? An alpha hemolytic microorganism that I have presented to you earlier. So that is Streptococcus, okay, Streptococcus pneumoniae. Okay, so umbilicate, so that's concave, depressed, at the center is an ini, okay. Umbonate, on the other hand, okay, it's raised, this one. This one. Okay, umbonate, it's raised, bulging center, it's outy. Okay, that's umbilicate and umbonate. So this one, umbilicate, okay, and this one, umbonate. Another thing you have, again, as what I have mentioned, the Streptococcus pneumoniae is umbilicate. Staphylococcus aureus, on the other hand, is convex. Okay, beta hemolytic streptococci are usually flat. So I know it would take a long time for you to really memorize that one, especially right now that the microorganisms are also changing. So you really have to do multiple testing before you identify correctly the species of the microorganism. Next one, also this is Bacillus cereus. Okay, umbilicate, this one. Okay, this one, this is umbonate. Again, this is umbilicate. How about this one? This is flat. And this one, this is pulvinate. Okay, pulvinate. Okay, another thing, the density. Okay, or the transparency, the translucency, and the opacity of the microorganism. So we have transparent, of course, you can see through the agar, okay, on the colony. Translucent, and this one, opaque. Okay, just like, but usually class, I'm telling you that based on experience, this is not usually observed, okay? What we usually observe are the size, the elevation, or the hemolysis. That's the most obvious. But for density, you really have to be very observant, be very keen, okay, in looking into the colonies for you to really say it's transparent, it's translucent, or it's opaque. Okay, so for example, beta hemolytic streptococci. Okay, again, I have given you examples of beta hemolytic streptococci, such as the Streptococcus pyogenes, okay, and Streptococcus agalactiae, right? So, beta hemolytic streptococci, of course, except the Streptococcus agalactiae, are usually translucent. Okay? S. agalactiae, as what I have said, although it is beta hemolytic, it's just semi-opaque. Again, I've mentioned this one, bullseye, because you have the medium-sized colony, and of course, you have here the hemolysis surrounding it. Staph or Staphylococci and other gram-positive and gram-negative microorganisms, they are opaque, and one of the unique characteristics of Bordetella pertussis is like a shiny, half pearl-like on blood-containing agar. Okay, I will let you see it later. Okay, the shiny half pearl. Okay, so that's Bordetella pertussis. Okay, anyway, here it is. Okay, so this is in a Reagan low agar. So this is a charcoal blood agar. So as what you can see, Bordetella pertussis, okay, is again shiny half pearl on Reagan low agar. Okay, aside from that, you also have the color. So the general description, of course, of the microorganism. So again, it can be white, gray, yellow, or buff. And I have told you earlier, okay, in the sample problem, that gram-negative microorganisms are usually colored what on blood agar plate? Okay, color gray, and they are usually large and mucoid. Another thing, okay, for Staphylococcus aureus, they can be creamy and buttery looking. So it's like white and yellowish. Okay, that's for Staphylococcus aureus. So here are the other examples for coagulase negative staphylococci. Okay, coagulase negative staphylococci, you have color white. Okay, enterococci, gray, just like your gram-negative. Okay, gray also on BOP. Okay, micrococcus and non-pathogenic Neisseria, they are usually yellow or off-white. And diphtheroids, they are buff. Okay, so this one. Okay, that's an example of the color, okay, the white color of coagulase negative staphylococci. Okay, micrococcus, you have here yellow or sometimes off-white. Okay, another thing, how about this? Again, large, gray, and mucoid, somewhat mucoid. So these are, again, gram-negative microorganism. How about this one? Consistency. So it can become brittle or splinters, creamy or butterous, okay, dry, waxy, and sticky. So these are the consistency. And of course, how do you determine this consistency is because, okay, how will you determine this consistency is by picking up or touching the colony using a sterile inoculating loop. So that's the only way you can determine the consistency, or maybe by just looking at it. Okay, and another one, okay, you have different examples here, just like what I have mentioned. S. aureus, that's creamy in consistency. Okay, Neisseria species, that's sticky. So meaning to say, when you touch the colonies with the inoculating loop, the entire colony adheres to the loop. Okay, Nocardia species, on the other hand, the colonies are brittle, crumbly, wrinkled like a breadcrumb. Okay, diphtheroids, they are dry and waxy. So when you push the colonies with a loop, okay, the colonies will remain intact. So that's dry. Okay, another thing, beta hemolytic streptococci, they are dry except of course for mucoid type. Okay? Another one, you have the pigment production, and one of the pigments that I have mentioned again is the pigment of Serratia marcescens, color red. So that is observable, okay, in particular at room temperature, the prodigiosin. Okay, so this pigment is an inherent characteristic of a specific organism in contrast to color. Okay, because for example, Serratia marcescens can be colorless on Mach concave. Okay, if you incubate it at 37 degrees Celsius. Because in the first place, your Serratia marcescens, okay, is a non-lactose fermenter. So, if you incubate it further at room temperature, that is where you can observe the unique pigment production. And in that sense, the Serratia marcescens becomes brick red. Okay, so that's pigment. So example again, you have Pseudomonas aeruginosa because of pyocyanin, and pyoverdine again, S. marcescens because of prodigiosin. Okay, of course you have the Cloivera species. Okay, that's color blue pigment, and of course very obvious. Chromobacterium violaceum, this is colored purple pigment. This is showing you the green pigment of Pseudomonas aeruginosa that is in Miller-Hinton agar. Another thing, you can also observe the greenish coloration of the pyocyanin and pyoverdine in the blood agar plate. And when you slightly open the plate, you can observe that there is a fruity odor coming from the culture of Pseudomonas aeruginosa. That's very, very distinct. Okay, you will get used to the smell if ever you are always culturing Pseudomonas aeruginosa. Another thing, this is again pigment production of Pseudomonas aeruginosa in Miller-Hinton. Okay, color green. Serratia marcescens, very distinct at room temperature again. Okay, another one. Okay, that's again on Mach concave agar. Okay, next one, this. Okay, so as what you can see, the margin is even, so it's entire. It's also umbonate. And again, there is red coloration, okay, because of the pigment prodigiosin. But this is in Mach concave. That's why you can see colorless here, colorless agar of Mach concave, because again, Serratia marcescens is a non-lactose fermenter. Next one, how about this one? There is black coloration. This is in XLD. Okay, this is deoxycholate citrate agar. Okay, so as what you can see, there is black colonies here. Okay, that is indicative of the growth of salmonella. Okay, for other, so we don't usually intend to smell the culture except if we are streaking or we are doing the workup and we can smell, okay, the odor of the microorganism. But here are the examples of the odors of the microorganism. So for S. aureus, it can be an old sock odor. For Pseudomonas aeruginosa, fruity or grape-like. Proteus, burnt chocolate. Haemophilus, mousy. And Nocardia species, freshly plowed field. So it's up to you really on your brain cells on how to recognize the odors of the microorganism. So usually, again, we don't intend to smell this. But when we open the plate and the odor is distinct, usually the odor will just disseminate. Okay, and do not inhale directly, of course, the culture. And of course, here are the examples of the growth of the colonies, okay, or of the bacterial colonies on tube media. So you have here thread-like growth, that is filiform. Okay, tree-like, so that is arborescent. Okay, beaded, very obvious. Effuse, it's spreading on the slant. Rhizoid, that's root-like. Okay, and echinulate, that's spiny, like also a vertebrae. Okay, another growth that you can observe is of course from a broth culture. Okay, so you can see growth, okay, diffuse throughout the broth culture, or the broth, I mean. Okay, the surface only, okay, bottom only, or subsurface. Usually this indicates microaerophilic microorganisms. This one anaerobic, this one aerobic microorganism, and this one facultative microorganisms. And another thing, okay, you can see in the book this puff balls, okay, that is usually indicative of the growth of Pseudomonas. Okay, but not only Pseudomonas, there are other microorganisms with that puff ball growth. Okay, so just like this one. So in number 1, as what you can see, there is the growth of obligate aerobes. Okay, so meaning to say, they have to really have enough oxygen to extract energy from their food. So that's why they will gather at the top of the test tube near the surface of the broth. Okay, where you can, of course, see maximum amount of oxygen. Okay, another thing, okay, as opposed to tube 1, we have here the tube 2. So that is obligate and aerobe. So usually, because they are killed by oxygen, they can be found at the bottom of the test tube, staying away from the oxygen. How about this one? Okay, you can find growth all throughout the all throughout the broth, whether at the surface, whether at the bottom or at the middle. Usually, these are facultative bacteria. They can live with or without oxygen. Okay? How about this one? Subsurface growth. This one. So again, as what I have mentioned, usually they gather at the upper part of the test tube, but not really at the uppermost, not really at the surface. Okay, so these are microaerophilic. So these microbes require only smaller amount of oxygen. Okay, how about this one? It's like the growth of facultative bacteria. Okay, but they are evenly spread. Okay, all throughout the broth medium. Okay, so these are aerotolerant bacteria. So meaning to say they are not affected by oxygen at all. So they spread evenly, okay, on the broth. Okay, so these are just examples, okay, of the true observation that you can have, okay, on broth culture. Okay, so that ends my discussion for colonial morphologies. I hope that after you have performed the isolation technique, you will use the techniques that you have learned here to really observe properly the microorganisms in order for you, of course, to properly identify the microorganisms. Thank you, class.